I have breast cancer.
I have breast cancer.
I have breast cancer.
And I needed someone to talk to.
I needed information.
I needed to know I could do this. I found Living Beyond Breast Cancer, an organization that understands what you need and need to know. They helped me, and they can help you too.
I have breast cancer, and I have support.
I have information.
And I have hope. I have Living Beyond Breast Cancer. Find 5 ways we can help you at 5ways.lbbc.org.
So there are some of the women that were in that commercial are here today. It was really fun to produce that. So I want to welcome our live stream audience to the Living Beyond Breast Cancer Conference. Welcome. And I want to remind everyone that if you are on Twitter, please feel free to tweet. Our hashtag is #ThrivingTogether, the number 2. So please let other people know what's happening here. For those of you that are not live stream but are in the audience, at 1 o'clock we're going to be signing a really important petition. I'll be making more announcements about that, but make sure you stick around to do that. And before I bring up our speaker, I just think it's always helpful to sort of know who's in the room. So I'm just going to try to do this really quickly. But for those of you that were diagnosed with metastatic in the last year, can you stand up so we can see those that are more newly diagnosed? And how about those in the last 2 years who've been working on this? All right. And 3 years? Do you have any 3? All right. And how about anyone more than 3 years? That's amazing. So I think for those of you that are here for the first time and you are sort of facing this in your first year, I hope this gives you a lot of comfort and a lot of inspiration. And I know everyone would be willing to talk to each other and share their stories. So thank you. So now I am going to introduce our keynote speaker. And we are really lucky that Douglas Yee, who is the director of the Masonic Cancer Center at the University of Minnesota, has traveled all the way to Philadelphia to share his knowledge. So please come up, Dr. Yee.
Thank you very much, and I actually want to thank the organizers so much for inviting me. I'm really honored to be here and to give you this presentation and talk. Although I didn't know that a pink tutu was the dress option, I would have done that. Yeah, okay. So I would like to talk to you a little bit about how we got to where we are. Maybe I should, I'm hearing a little ringing. If I shut that off, am I okay? Okay. How we got to where we are, and I'm going to use a set of slides I usually use for medical students. And I will guarantee you, so I teach in the medical school, I'm a medical oncologist, I take care of breast cancer patients, I also teach graduate students. I will guarantee you that women with breast cancer know a lot more about breast cancer than medical students, so you should not be intimidated by these slides. So what I'd like to do is talk a little bit that how did medical therapy develop for breast cancer. So I want to take a little bit of a historical view and try to bring you up to the future very quickly. So how did we learn about breast cancer? And the first couple of examples I'm going to give you around estrogen receptor, and I will try to show you that was really a very lucky guess that estrogen receptor had anything to do with breast cancer. We'll talk about HER2 and how that actually came out of a very important clinical question. We'll say a few words about triple-negative breast cancer and how that really sprung from our new technology and our ability to classify breast cancer, and say a few words about what's next. But I hope you will ask me the what's next questions at the question and answer session. So this is a paper from The Lancet in 1896. It describes Dr. Bateson's example of how you can treat inoperable cases of the breast. So if you ever have time, The Lancet actually has done a very nice job of archiving all their old literature, so you can actually pull this out as a PDF file. But it basically discusses how Dr. Bateson, who was a surgeon, was confronted with a young woman with a locally advanced breast cancer who was inoperable. Then when you read this paper, he digresses into a very long discussion about, well, he was an apprentice on a Scottish farm, and he was following the shepherds around, and they used to take the ovaries out of their animals, particularly their sheep, to change their lactational cycles. So then he figured out, well, the breast had something to do with the ovaries. So he reports in his paper, I'm just going to take the ovaries out of this woman. So that one must have been tough to get through his institutional review board, but he did it, and the breast responded. So it is at a time when nobody, so first off, the importance of this particular observation was, it was the first time the surgeons actually figured out you could do something for cancer other than operating on the cancer. And it was also done at a time when no one really had any understanding of estrogen, ovarian function, of the endocrine system, the estrogen receptor. So fast forward about 100 years, and we have a little bit of better understanding of estrogen receptor function now. So this cartoon, and I know we're on both sides. I'll try to do my equal pointing on one side or the other, but I'm going to go this way first. This cartoon is meant to be the nucleus, and this is the estrogen receptor. So we have learned that the estrogen receptor binds, once it's bound to estrogen, binds to DNA in the nucleus and initiates a program of gene transcription. So in the absence of the estrogen, the estrogen receptor doesn't do a whole lot. In the presence of estrogen, the estrogen receptor kicks off its programmed set of operations. And the reason we do have that is because women evolved to have lactate every single time they ovulate. In other words, you were designed to have a child every time you ovulated. So in the first part of the menstrual cycle, estrogen goes up, and then if an ovum isn't fertilized, it goes back down. So it's a signal for mammary cells to grow, and then if estrogen was withdrawn, is to die. As it turns out, some breast cancers remember that, and we've learned that how it works that through this through this nuclear estrogen receptor. So the global we developed a lot of different drugs to target that. The selective estrogen receptor modulators, shown here, tamoxifen being the first, bind the estrogen receptor and keep it in an inactive complex. So in other words, it serves as a cap to allowing estrogen to bind the receptor. So for premenopausal women who might have estrogen receptor positive breast cancers, this is the only drug option if their ovaries are intact. Dr. Bateson did this. He took out the ovaries of women, and we still do that as a useful tool. We also know that another non-surgical way to block ovarian function is to give the LHRH antagonist. And that's been very effective as well. Now I'll go over to this side. So on this side, we also know that postmenopausal women can still make estrogen from adrenal precursors. They're converted by a molecule called aromatase, and aromatase is blocked by multiple different agents. So this particular endocrinology that is important in breast cancer and ER-positive tumors was displayed by Dr. Bateson well over 100 years ago. Just also as an aside, anytime I say something that ends in the three letters A's, means it's an enzyme. Enzymes catalyze biochemical reactions. Most enzymes are targetable by drugs. So most of the drugs we have target enzymes that are important in this pathway in the different regulation. So as it turns out, the estrogen receptor can be blocked and activated by a lot of different pathways as well. So we just said a few words about this particular pathway binding estrogen, going to the nucleus, inducing a gene transcription program. But it turns out cells can also have similar activation pathways through multiple different molecular pathways. As it turns out, estrogen receptor can initiate gene transcription even in the absence of estrogen. So in this particular example, IGF receptor and EGF receptor can signal to this estrogen receptor. And even in the absence of estrogen receptor, it can initiate transcription. So molecules in this pathway, particularly the molecule mTOR, was the first molecule where we decided we're going to make an enzyme that blocks that particular a drug that blocks that particular enzymatic pathway. So shown here is a cartoon that's very similar, and the first drug approved for breast cancer, in addition to the ones I showed you on the previous slide, was everolimus or Afinitor. Afinitor blocks a pathway called mTORC1, mTOR is a kinase which phosphorylates other molecules, so everolimus or Afinitor blocks that particular activation. These are the clinical trial results, and I don't know that if many of you are used to looking at clinical trial results, but I want to show you what happened when we did these studies. So there were 2 trials that were done with everolimus in the beginning. The United States did a trial in combination with exemestane, the aromatase inhibitor, and it shows you a Kaplan-Meier curve where basically patients are started on the drug and they get the control drug, in this case exemestane, with the drug everolimus or exemestane alone, and then we measure basically how long can you stay on the drug. As it turns out, patients who got the combination stayed on longer than patients got exemestane alone. I know for some of you that might have been on this drug, it's not the easiest drug in the world to take, so it might have been longer, but it might have felt like a lot longer. But that's a challenge for any drug development. I wanted to show you the other side too, because it points out a question that we don't understand very well. Our European colleagues did the trial with tamoxifen, and they paid attention to what they call primary resistance or secondary resistance. In other words, some women have the estrogen receptor and have not responded to a strategy to block the estrogen receptor. We call that primary resistance. The definition of primary resistance in the clinical sense is if you start on a drug and it doesn't benefit you for more than 6 months, that's primary resistance. When our European colleagues did that, they then showed that patients with primary resistance or tumors that have the estrogen receptor but never responded to an anti-estrogen don't appear to benefit from the addition of everolimus as opposed to patients who had secondary resistance. So to me, this remains an unsolved problem, why some tumors appear to have the estrogen receptor and don't care a whole lot about it. And we'll come back to that in a little bit as well. So when we come back to this cartoon, this little arrow, I'll go to this side for a while, we'll go to this little arrow, and it says cell growth. So cell growth is something that's stimulated through the MAP kinase pathway. I will say there are several MAP kinase inhibitors, or particularly ERK inhibitors, MEK inhibitors working through the pathway, but cell growth is the other thing that stimulated by these other pathways. So what modulates cell growth? This is a cartoon that shows you the cyclin-dependent kinases, another A's word, 4-6. So one of the things I want to comment on, and I'll come back to this at the very end, all science is important, and the reason I'm saying that is because the cyclins were discovered by basic scientists who are just interested in how frog eggs actually divided. So if you think about a frog egg, they're big, they're easy to isolate. After they're fertilized, how do they continue to grow? And our colleagues in basic science discovered there's a series of kinases, enzymes that modify other proteins called cyclins, and to get the cell to start making DNA and start making cells to divide. I think as oncologists and as basic scientists, we're very opportunistic. If the frog guys or the fly guys or the worm guys show us something that's relevant to what we are interested in, we're more than happy to take their data and see if it works for us. So at any rate, this cartoon shows that the cyclin-dependent kinases interact with cyclin D1. Cyclin D1 then, once it's released from the cyclin-dependent kinase, they go on and phosphorylate the protein called retinoblastoma. The RB gene is a gene that's deleted in children with retinoblastoma. That's another discovery story that's kind of interesting. And phosphorylated RB then allows cells to go through the cell cycle. So if you think about this pathway, then why don't we make an inhibitor to the A's? So the A's, the cyclin-dependent kinase here, is a perfectly good drug target. So this demonstrates the first drug that was approved, palbociclib, and this is in combination with fulvestrant. Again, the similar Kaplan-Meier curve showing you the patients who received palbociclib fulvestrant stay on the combination longer than patients who only got fulvestrant. And I should have mentioned all the trials I've shown you so far were trials that were done in patients with metastatic disease. As you know, and I noticed there's a display outside. The second drug was approved, ribociclib, based on these data. These are data that were previously untreated, and they got ribociclib and an aromatase inhibitor or the aromatase inhibitor alone, and there was a better result. So I wanted to show you these couple of things with ER, both the mTOR story, in other words, the everolimus story, and the CDK4/6 inhibitor story, because it is a landmark in how we treat ER-positive disease. For the 60 or so years since we've had tamoxifen, we've never been able to show anything combines with tamoxifen or just blocking the ER. So now that we know we can block estrogen receptor and we can block other pathways, that opens up the door, in my mind, to more discovery and more potential options in the future. So what's next for the estrogen receptor? I think there will be more anti-estrogens. There will be some oral fulvestrants. For those of you that are on Faslodex or fulvestrant, you know that shot is not a whole lot of fun. So there are some oral drugs that are coming. Inhibitors of other pathways. I actually have to leave the room when they give that shot. That's a gigantic shot. There are other pathways. I said the everolimus targets mTORC1, which implies there's an mTORC2. So there are inhibitors of TORC1 and TORC2 coming forward. If you were paying attention to the pathways, there are molecules both up and downstream from mTORC1, and those inhibitors are coming as well. CDK4/6 also implies there's CDK1 through 8, and those pathways may be relevant in breast cancer at the same time. So I think those are all coming forward, and I'm looking forward to seeing how they develop. So I'm going to talk a little bit about HER2 and try to get the understanding of how HER2 was actually discovered. So in the 80s, a variety of investigators, including one of my mentors, the late Bill McGuire, was interested in trying to understand how women with absolutely identical tumors had different outcomes. So Dr. McGuire kept a very large tumor bank, and one of the first things he did was determine tumors that had high S phase. In other words, tumors that were rapidly dividing. So S phase is a term of art. When we think about the cell cycle, cells are in G1, then they go into S phase. S stands for synthesis. They're making DNA. Then they go into G2. Then they go into M and mitosis. So they basically make DNA and then they have to split. So one of the technologies developed in the 80s was to determine how many of the cells in a breast cancer at any given time were in S phase. And as it turns out, high S phase tumors had worse outcomes. So then the next question, obvious question, I think, is what is driving S phase? Because if you knew what was driving the DNA synthesis pathway, then you would have a better understanding of what we should be targeting. So this is where HER2 was first discovered. And HER2 was an interesting confluence of events of a couple things at the same time. First, in a tumor called neuroblastoma, which is a neural tumor mostly of children, but this is a rat model, we're using an assay and new was identified. When I say using an assay, I'll tell you what the first discovery pathways were. If you put normal cells on a dish, they will stop growing when they contact each other. If cells are transformed or become cancer, they'll now start piling up and form little focuses. So Dr. Weinberg discovered new. He basically took a rat neuroblastoma, ground it up, put the DNA on a cell, a normal fibroblast, and then little piles that would form, he'd yank it out and figure out what was in it. And it turns out it was the gene he called new for neuroblastoma. So if you ever heard the term HER2 new, that's where the new comes from. New is related to the epidermal growth factor receptor superfamily. It was the second member. The first one, EGF receptor, was already discovered. We'll talk about HER3 and HER4 in just a second. But that was good because as a growth factor receptor, it does what its name implies. It stimulates growth. So maybe this could be an explanation for high S-phase tumors. It also was related to a viral oncogene. Chickens can get a funny kind of red cell leukemia called erythroblastosis. And I'm not going to go too deeply into why chickens get cancer, but as it turns out, chickens get cancer because they take a little of this virus takes a little bit of their genome, truncates it, and gives it back to them. The reason the virus does this is because the virus wants to survive. It doesn't care that much about the chicken. And it got its other name, ERB-B. So you will hear HER2 ERB-B all talking about the same gene. And they are growth factors that bond multiple ligands and stimulate transmembrane tyrosine kinases. So another A's word suggesting targeting is possible. Dr. McGuire collaborated with Dr. Slayman in the same tumor bank to show that if tumors were amplified in HER2, the outcomes were more aggressive. This is more than 5 copies, showing you in a very small subset of women with more than 5 copies, and I believe these were mostly node-negative patients, had enhanced time to recurrences. So it fits the story that maybe this is something that is driving these cells, and we don't do, we now measure HER2 by other assays. At our institution, we tend to use fluorescent in situ hybridization, or FISH. The little orange dots represent HER2. The little green dots represent the center of the chromosome it's on. So you can see in some of these cells, they really have a lot of HER2 compared to the copies of the chromosome they have. And you know, my belief in cancer cells, the cancer cells are very efficient. They don't bother to do something or spend their time on something unless they really need it. So this was an argument they really need HER2. So when the first monoclonal antibody was created, trastuzumab or Herceptin, it was tested in women with metastatic breast cancer. So first, the trastuzumab is shown here. The little yellow dots, the little yellow areas, are areas that were identified from a mouse. So the first anti-HER2 antibody came from a mouse, because mice are easier to make monoclonal antibodies. But you can't get injected with a mouse antibody. You'll develop an antibody to the mouse antibody. So trastuzumab was modified into the gray parts. These are all a human immunoglobulin. And to be straightforward, HER2 was first discovered as a diagnostic test, or the antibody Herceptin was first discovered as a diagnostic test, in the idea that, okay, well, gene amplification is important. Let's just see if we can measure it by looking at protein in an immunohistochemistry slice. But as it turned out, when you dumped the mouse monoclonal antibody on cells that were amplified for HER2, it killed them. So that was an interesting process. Dennis Slayman was really key in making sure this moved forward. And this was the first clinical trial demonstrating, again, that the benefit of chemotherapy plus trastuzumab or Herceptin was better than chemotherapy alone in patients with metastatic disease overexpressing HER2. So that was a lesson from HER2. It only worked in HER2 overexpressing or amplified breast cancer. It had significant synergy with chemotherapy in metastatic disease. And one of the things we did not understand at the time HER2 was developed is a congestive heart failure in combination with anthracyclines. This trial actually allowed patients to get either paclitaxel, Taxol with trastuzumab, or AC, Adriamycin cytoxan with trastuzumab. And this is how we discovered that the AC plus trastuzumab was particularly cardiotoxic, and that remains as a challenge for most HER2 positive drugs, targeting drugs. So HER2, because it's 2, suggests there's a HER3 and a 4 and a 1, and this is the little cartoon here. As I mentioned, HER1 is EGF receptor. This is HER2, this is HER3, this is HER4. And the way we think most of these family members work is they find a buddy. So they dimerize with another molecule, and then they actually activate their biochemical function, their kinase signaling through this interaction. So that then gives you a couple of thoughts on how you could particularly block that pathway. One way to block it is with an antibody. So that is what trastuzumab showed us. So trastuzumab binds HER2, and it inhibits signaling. The second drug that was created was pertuzumab. Pertuzumab, or Perjeta, also binds HER2, and it prevents this HER2-HER3 dimerization from occurring, also blocking signaling. As you know, trastuzumab and pertuzumab are given together now. And then there's TDM1. So TDM1 is a very interesting molecule. It's the antibody drug conjugate. So it is trastuzumab with a little chemotherapeutic agent stuck onto it. And the idea is that it gets internalized and releases this chemotherapeutic agent. I think antibody drug conjugates are just really fascinating, and we have a really fabulous opportunity to work on antibody drug conjugates. I'm not going to say too much more about them today at this talk, but please ask, because I think this is a very interesting way for anybody who's had TDM1 or Kadcyla It is certainly less toxic than giving Taxol, Herceptin, and Perjeta, and that's an important goal for treatment of all women with breast cancer, but particularly in the metastatic setting. So I want to introduce this, and we'll talk a little bit about immune therapies in the next session if you choose to come to the breakout. But it is an antibody, so trastuzumab is an antibody, and we have antibodies. We make antibodies for a reason. The reason we make antibodies is to initiate an immune attack against the thing the antibody is directed against. So if we break down the structure of trastuzumab a little bit, here's the leftover mouse chunks that bind the HER2 molecule. Here's what we call the FAB domain. Here's what we call the FC domain. This is a model of a tumor cell expressing HER2 and a white blood cell. So the idea is that HER2 is bound to trastuzumab, but the little FC domain, the little chunk that was taken from a normal human immunoglobulin chain, can now bind white cells. And potentially, the white cells can initiate an immune attack against the tumor. I'm not going to show you the data, I'm not going to show you the data for why that is very interesting, except to tell you this is a pathway that's being moved forward in HER2-positive tumors. So when we think about HER2, we want to present dimerization with other partners. We know the drug pertuzumab. There are several HER3 antibodies that are in clinical trial now. We can inhibit the kinase activity. So we didn't talk a little about lapatinib or Tykerb, but certainly that's been approved for several years. The other drugs, neratinib and poziotinib, are also coming very soon. The antibody drug conjugate, I mentioned Kadcyla or TDM1. And I think, to me, a very interesting aspect is modified antibodies that have an FC domain that is more likely to interact with white cells. Will they actually be more potent in initiating an immune attack against HER2-positive cells? And I think that's an area that we should all pay attention to. So let's talk a little bit about what happened in the past couple of decades, is that we have begun to establish the idea that breast cancers are not all the same. Now, we already knew that, right? So we already knew they're ER positive and ER negative. We already knew they're HER2 positive, HER2 negative. But this is adapted from Dr. Peru, Chuck Peru at the University of North Carolina, initially at Stanford, then at the University of North Carolina, really pioneered the way we can begin to characterize breast cancer specimens. So every cell in your body has the capability of making about 30,000 genes. Now, obviously, by making 30,000 genes, I mean making the messenger RNA, turning that into protein and expressing it. Now, you know that none of us all express the same genes at the same time. Otherwise, we'd be a single-cell organism like an amoeba. The genes that are expressed in our skin are vastly different than the genes expressed in our muscle, which are vastly different than the genes expressed in our mammary epithelial cells. So in the 2000s, we began to be able to characterize this. I'm not going to go too deeply into technology, but breast cancer cells don't express all 30,000 genes at any given time. I think this array has roughly 700 genes on it, and I want to just show you what this is in case you haven't seen it before. So every column is a breast tumor. So each breast tumor is here, and across every row is a different gene. So if you really were getting up to the screen and look at this, first thing you'd say is, none of these tumors are identical. They're all slightly different in their expression profile, but you can begin to sort of cluster them. So there's the sort of green over here and red over there, and green over there and red over here. Green and red are just different degrees of gene expression. So high tends to be red, low tends to be green. And you can begin to cluster them, and they cluster into groups that we already knew about. So there's HER2 enriched, there's the luminal subset, which are actually the estrogen receptor positive subset, there's Claudin low, there's basal-like. So moving forward with that kind of technology, we actually discovered that things are not always what they seem. I want to show you this data from Lisa Carey. This is a neoadjuvant trial. So a neoadjuvant trial is women who get their chemotherapy prior to surgery. But Dr. Carey and her group, along with Dr. Peru, characterized what these tumors were. So this was a HER2 neoadjuvant therapy with trastuzumab. So every patient had to have HER2 gene amplification. So that was a given. When you begin to profile along these different pathways, if you look at, if you look at the blue, those are the HER2 enriched by gene expression profiling. So even though they all are expressing HER2, they're not all necessarily showing the activated pathway. Many of them were luminal A, and the luminal A are the subset that are estrogen receptor positive and HER2 positive. This argues a little bit they are actually more like estrogen receptor positive tumors, and others were also mixed in the subset. If they then looked at the responses to the HER2-based therapy, they found that only the HER2-enriched subgroup substantially benefited over chemotherapy alone. So I want to point this out to you because even though we think we're smart enough and we can say, oh yeah, this is a HER2-positive tumor, you should benefit from HER2-positive therapy, I think there are deeper dives we need to do into tumors to try to predict specific individual responses. However, the biggest challenge has been with triple-negative breast cancer, and I think the gene expression profiling has really started to illuminate triple-negative breast cancer. So I will first say, and apologies to the triple-negative breast cancer advocacy group, I really dislike the term triple-negative breast cancer because a couple things. One of them, for me, it displays our ignorance in being unable to classify what it is. I don't really like to classify something what it is not. So what it is not, it doesn't express ER, progesterone receptor, or HER2, but it is something. So anyway, that bothers me. And the second part about the triple-negative breast cancer, it does make an assumption they're all the same. And I hope to show you in the next couple of minutes, they're really not all the same. And that's a challenge for us going forward. So in 2011, we just call them triple-negative breast cancer. This is the work of Jennifer Peyton-Pohl at Vanderbilt. She was very kind to share these slides with me. Dr. Peyton-Pohl started the gene expression profile triple-negative breast cancers. And in 2011, she identified 6 subtypes. She has subsequently modified that to a more recent in 2016 to show it the 4 subtypes. The biggest subtype that disappeared is the immune subtype, or the IM, shown in black here. So when we do gene expression profiling, we take a tumor, we grind it up, we take a look at the RNA, and we say what it is. So anything passing through that tumor at any given time is captured. So the immune phenotype was not coming from the cancer cells, it was coming from a lot of immune cells that were contained within the tumor. So when Dr. Peyton-Pohl did a very deep dive into what these were, she identified that particular subtype, the immune phenotype, was not a feature of breast cancer, it was a feature of the tumor. So just to make sure I'm clear on the difference. We want to know what the breast cancer cell is doing, but the tumor itself is more than just the breast cancer cell. It's blood vessels, it's connective tissue, and in some cases, it's a lot of immune cells. So, and I know this is hard to see, but this was Dr. Peyton-Pohl's dive into what immune cells look like. And I just want to show you that many of the cells had lots of lymphocyte infiltration. It is hard to see, and if you want to get the paper, it's a little easier to see, but at high power, this tumor is almost all lymphocytes. There's not that many epithelial cells. This tumor has very few lymphocytes, and when she characterized it, she could identify tumors that had high lymphocytes and those tumors with low lymphocytes. And her immune signature prior to this year were those cells with high immune infiltrates. So first, when she began to say, well, where did the immune signature go? It was spread across several of the other subtypes. But it raises an important question, is, why were they there? Why were they there in some cells and why weren't they there in other cells? So the next description I'm going to give you is sort of, I think, compliments of Judy Erdahl when I explained this to another group. She said, oh, that makes sense to me. So at any rate, does anybody not know where this comes from? If somebody has not seen Star Wars, I just don't know what I can do. But anyway, this is the famous scene where Obi-Wan, Luke, R2-D2, C-3PO are in their speedster and they show up in town and they're trying to find Han Solo. But at any rate, the stormtroopers are out there looking for the 2 droids. So in my analogy, the speedster is the cancer cell and the stormtroopers are immune cells. So the immune cells are actually trying to find these cells, but find the 2 droids, the 2 features of this particular speedster that they should recognize. So then Obi-Wan does the Jedi mind trick and says, these are not the droids you're looking for. So even though the immune cells are there, they can't recognize these as different because the cancer cell is doing the Jedi mind trick. The cancer cell is saying, don't look for this cell. And if you come to the immunotherapy breakout, and if you don't, just watch it because I guess it will be online. We'll talk about this in a little more depth. But we don't want our immune system to start attacking our normal cells. We want a way for our immune system to recognize self from not self. So cancer cells will learn the self, to disguise themselves as self, and they do that through a variety of mechanisms. One mechanism is through checkpoint inhibitors. So checkpoint inhibitors, anybody see USA Today yesterday? So USA Today had a relatively, through my eye, it was kind of a negative overview of some of the new drugs, the checkpoint inhibitors, but in particular, the checkpoint inhibitor pembrolizumab, which was approved in lung and melanoma, was a feature of that particular article. But pembrolizumab is the way that we try to overcome the Jedi mind trick of saying these are not the cells you're looking for. So pembrolizumab is an antibody that binds a specific receptor, I always get them mixed up between the ligand and the receptor, but blocks the PD-1, PD-L1 interaction. It was approved in, as I mentioned, breast and non-small cell lung cancer, and Rita Nanda has done the phase 1 trial showing that there are patients who have triple-negative breast cancer who respond very well. So these are little racetrack plots. This patient in yellow demonstrates somebody who had a partial response, but it's ongoing, and it's ongoing in this particular slide for 64 weeks, which is pretty good for stabilization of disease. So there have been good responses, but at the same time, there have been responses that have been not so good. So if you, again, assume all triple-negative breast cancer is the same, maybe this is the result you're going to get. You really have to find the cells that have been able to track, the tumors that have been able to track some features of the immune system, but can't quite activate them. And I think we'll have to do a better job at classifying triple-negative breast cancer in the future to be a little bit better. So what are the other subtypes? So the LAR type is luminal androgen receptor. So these tumors express the androgen receptor. And fortunately, we know a lot about the androgen receptor from all our colleagues who are doing work in prostate cancer. So even though we call these triple negative, they are positive for something, and they are positive for the androgen receptor, and whether or not they respond from androgen receptor blockade will require some work. The basal-like 1, we'll say a little bit more about, but those are tumors that seem to have DNA damage response genes as driving their phenotype or their genotype. Mesenchymal are tumors that have growth factor signaling, and basal-like 2 have specific growth factors, the MET receptor and EGF receptor. So just think about this for a second. If you were going to do a clinical trial where you were going to block the androgen receptor and you decided you were going to include all triple-negative breast cancer specimens, patients, you would really have to be very careful to pay attention that only 16% of the disease we call triple-negative breast cancer might benefit from your drug because only those express the androgen receptor. I think we, as a group, are getting smarter about moving those kinds of trials forward. But I think it's a very important thing we need to recognize, and where I started, why I don't like the term triple-negative breast cancer. I'd rather really say you have androgen receptor positive breast cancer, or you have DNA damage repair positive breast cancer, and that means something in terms of therapy and outcome. So I do want to say a word about the DNA damage repair pathway. We have known for over a decade now the idea that poly-ADP ribose polymerase, another A's, shorthand PARP inhibitors, work in patients with BRCA mutation carriers. Dr. Alan Ashworth really pushed this concept forward, and the clinical trial was reported in 2009. How do PARP inhibitors work in BRCA-dependent tumors? So this is really the question about what's driving DNA damage repair tumors. So BRCA family members have inherited a defect in the DNA repair pathway. And the principle behind PARP inhibitors is you take out another pathway. It's kind of like a five-legged stool. So if there are 5 stools of DNA repair, your tumor already has one out, you want to kick out the other. This is explained as a synthetic lethal concept. So this is shown on this part of the slide. The synthetic lethal thing is you have two pathways intact, you're fine. If you have only one pathway out, like BRCA1 mutant patients only have one pathway out, you're fine. If you have the other pathway out, you're fine. You have to take both of them out before you get cell death. I don't want to go too deeply into what the PARP inhibitors do, but this is PARP, poly-ADP ribose polymerase. It binds DNA at single-stranded breaks. It will find a single-stranded break, and it will fix it. It will fix it, and then it requires homologous recombination, or the thing that BRCA1 patients do, and BRCA1 patients deficient in, and so you have this whole pathway. If you then come in with an enzyme that blocks the ability of the complex to leave the DNA, you need HRR, homologous recombination repair, or the thing that BRCA mutant tumors cannot do to fix that, and that's why these are synthetic lethal. So one leg is out, the other leg is out, cell dies, and that's the principle behind the PARP inhibitors. So BRCA tumors lack homologous recombination repair. So you probably also know that olaparib, the first one, was approved in ovarian cancer with germline BRCA mutant tumors. It's a pet peeve of mine, actually, why this wasn't done in breast cancer at the same time, but it wasn't. And it has to do with numbers of patients. But at the same time, in March, just recently, olaparib increased indication for patients with ovarian cancer, and I think approval for the PARP inhibitors in breast cancer will be coming very soon. But again, it's going to be a challenge for us to go beyond just BRCA mutant patients as to whether or not there's a larger group of the triple negative subgroup, particularly the basal-like one, that would benefit. So I'm going to end my last couple of minutes on why do breast tumors stop responding. This is kind of my pet peeve. Breast tumors are caused by accumulated genetic abnormalities. We don't think, and this is debatable, but we don't think, it's not like lung cancer, melanoma, or exogenous mutagens, tobacco smoke, or sunlight are linked to the majority of breast cancers. They may be linked to some, but certainly not the majority. As it turns out, we have our own source of mutation and are frequently expressed in breast cancer. So I didn't think of this at all until I contacted my colleague, Ruben Harris. Dr. Harris has been working on the cysteine deaminases for a long time. And honestly, I didn't know this, but I should have known this, because if you think about it, we have to have the ability to mutate our genome endogenously. In fact, our immune system has to do this all the time. If you get exposed to new antigen, you have to make a new antibody. So you have to shuffle your genome to make a new antibody to the thing you're exposed to. One of the very critical molecules to do that is the ApoBac family. There are 9 active enzymes in humans. They're conserved across multiple subtypes. I just want to show you the idea that one of them, ApoBac3B, is the one we're very interested in. So this is a C, and ApoBacs catalyze that into a U. In other words, this is a mutate, and then in DNA that gets changed to a T. And this is a mutational pathway that a lot of mutagens do, and we all have it. We all express the ApoBac family members. As it turns out, some breast cancers express more ApoBac3 than others, and this is a paper we published a couple of years ago now in Nature, outlining the fact that ApoBac3B is an enzymatic source of mutation in breast cancer. In other words, these are breast cancer cell lines, but they're also true of breast cancer tissues. Many breast cancer cells are expressing high levels of this endogenous mutagen. Does it make a difference? This is a mouse study where we treated mice with tamoxifen, and the levels were either high or low. And the high-level tumors in mice actually responded to tamoxifen for a little while, but then stopped much sooner. In animals that had their A3B levels knocked down, tamoxifen response was longer. So I got excited about this when we worked with Dr. Harris, with the idea that I know, and many of you also know, that some women stay on therapies for very long periods of time. I saw a patient with triple-negative breast cancer who's been on Xeloda for 4 years now and seems to be tolerating it very well. I don't really know why that happens, as opposed to the woman who stays on Xeloda for 6 months and then progresses. We're hoping that maybe it's the A3B molecule. One way to show that is in combination with our colleagues in the Netherlands. Dr. Martins had a cohort of patients who were first treated with tamoxifen, and he measured how long they can stay on tamoxifen. And as it turns out, if you had low APOBEC levels, you could stay on longer than on high APOBEC levels. So that could be a very important pathway. It's not a pathway that's going to result in cell death, but it may be a pathway that will result in cells stopping accumulation of genetic mutations and optimizing current therapies. So what have we learned about breast cancer? I hope I showed you basic research fuels new ideas. New ideas are translated into new therapies, almost always first evaluated in metastatic breast cancer. We know now it is many disease. In many ways, breast cancer is becoming an orphan disease because of all the subtypes. But we can identify the fuel, which would argue the estrogen receptor, the driver, such as HER2, and Achilles' heel may be the mutational spectrum that we haven't fully understand to identify those who will respond from immunotherapies. And I would argue that stopping tumor evolution could enhance duration of response to existing therapies. My second to last slide, I'm going to take the bully pulpit here for a second. As you know, our federal government didn't shut down yesterday. It's going to shut down next week, maybe. But this is critically important to the research community. I want to make sure I'm very clear on this. I am trying not to take a political stance on this, but the president's budget has cut substantial amounts of money out of the NIH, $5.8 billion in the next upcoming year. The whole institute of the National Cancer Institute, the total budget is $5.2 billion. So this is essentially a required cut more than $5.8 billion. In addition, in this fiscal year, there's a proposal to decrease another $1.2 billion. So, like I said, this is not a political argument, but I hope I have shown you how science is important for whatever we do in healthcare. And if you have an in to your congressman, your representative, your senator, you should make sure you advocate for continuing what we call robust, predictable, and sustained increases in funding for research. So that So this is my favorite Gary Larson cartoon. It says, knowing how it could change the lives of canines everywhere, the dog scientists struggled diligently to understand the doorknob principle. So I actually believe we have many of the tools we need to solve many problems associated with breast cancer. We just need to have the support to do it. And I want to thank you, certainly thank you for your attention. One of the things that's very clear is we don't make any steps forward unless we partner with the patients we see around clinical trials, but just around education and advocacy. Scientists, clinicians, I think our elected officials are going to be extraordinarily important in the way science goes forward, and advocates as well. So thanks, and I'm looking forward to your questions. Oh.
Thank you so much, Dr. Oh, thank you. Thank you so much. I have to say that, you know, when we started this conference 11 years ago, this talk would have been a lot shorter.
Yeah.
So there really has been a lot of changes. There's a lot, I think this was very hopeful. I know it was a lot to take in early in the morning. I'm super proud of everybody. So we're going to take questions, and you can text them in. This is how you text them. Our live stream audience can text the questions too. So Janine and Kathy will be asking you the questions.
Okay.
Thanks, everyone. Well, while we're polling for questions, we do have a couple people who jumped the line already. So our first question, Dr. Yee, for someone with estrogen receptor positive breast cancer, if their cancer stops responding to Ibrance, is ribociclib or I don't know how to say the name of the other one, but are either of those options for that person?
Yeah, so that's an unknown right now. Just to comment, what's a parallel observation? So you know there are 3 aromatase inhibitors, anastrozole, letrozole, and exemestane, and they're not completely cross-resistant. And what I mean by that is you can take one, and if you stop responding, you can benefit from another. So it is unclear whether any of the CDK4/6 inhibitors work in the same way. Up until a couple weeks ago, all we had was ribociclib and palbociclib. Ribociclib was approved in the setting of first-line therapy, so patients who were previously untreated. So we don't really know the answer to that question. I will tell you, I'm interested in trying to understand the answer to that question, and it may be an option for women who have progressed through palbociclib.
Thank you, Dr. Yee. So our second question is about HER2-positive breast cancer. What is being done for those whose cancers are HER2-positive, but they have a low FISH score and don't respond well to Herceptin or Kadcyla?
Yeah, so first let me talk a little bit about a controversy about HER2 testing. In the adjuvant trials, it turned out some women with low HER2 testing still benefited from Herceptin. They were included in the trial sort of through just the way the trial was designed. When the trial for HER2 in the adjuvant setting was designed, women who were deemed to be HER2 positive by their local institution were given Herceptin, and then all that material was centrally reviewed. Under central review, there's a significant number of women that actually did not have HER2 overexpression or amplification, but they still were involved in the trial. And it looks like HER2 still benefited for those subsets of patients. So how do you explain that? So one way to explain that is through the idea that HER2 is an immunotherapy, that for you to respond to HER2, you may not need very high levels. You just have to have a very good immune response against Herceptin. And it has nothing to do with levels, it just has to do with presence or absence. So point number one, for a research question, I think if there's an opportunity to participate in one of the clinical trials that are using the modified HER2 antibodies like margetuximab, you look for that opportunity. In the world of standard care, if the patient has not had a tyrosine kinase inhibitor like Tykerb, that would be an option as well, because you would hit the same pathway through a different strategy.
Thank you. So, I'm sorry, we're getting a number of different questions here. So, my next question is whether there's a difference between using letrozole and fulvestrant in conjunction with Ibrance. Is one more effective than the other?
Yeah, I don't think that we have a very good answer to that at all. It happens to be the way the trials were designed. My personal practice is if somebody had been on an aromatase inhibitor, had never been on fulvestrant, then to switch it over. But it is not clear.
Thank you. So
I got to get an answer I can actually have a definitive answer to. Somebody's got to ask me something I can say.
we like to ask the challenging questions that don't have definitive answers. So this is the next question. Is the overall vision for current research progression-free survival while on treatment, or are we bold enough to be able to think there will someday be cures?
So, I am really intrigued by the immunotherapy part of the research. So I'm going to flip back for a second. If you didn't read the USA Today article yesterday, it's worth taking a look at. It really is about the new checkpoint inhibitors. The new checkpoint inhibitors have an overall response rate that is not dramatic. It's good, and it's good in diseases where we had nothing like melanoma. But if you take a very good close look at those studies, some patients with advanced melanoma or advanced small cell lung cancer have extension of no apparent evident tumor for very, very long periods of time. I happened to meet one of those patients a couple years ago. I participated in the Rally for Science at Capitol Hill through the American Association of Cancer Research. And she had been without evidence of disease for 3 years. She was one of the first patients on the non-small cell lung cancer trial. So there is a minority of patients who have what every oncologist would have said was completely incurable disease. They have no evidence of disease for extended periods. I think that's an exciting area. I don't think we're smart enough to know yet how to do that for everybody. Just to comment about breast cancer, in breast cancer, the current prevailing thoughts are that it only works in triple-negative breast cancer. And the reason it only works in triple-negative breast cancer is because, like I showed you on that little slide, those particular breast cancers might have more mutations. So they might look more like a melanoma or a lung cancer than a luminal A estrogen receptor positive breast cancer has very few mutations. So there's nothing for the immune system to recognize as foreign. So in those settings, unleashing the immune system to find out, waving off the Jedi mind trick in those settings will not work because there's nothing to recognize. And anyway, the next part, I'm going to talk a little bit about how we might improve that. But I really do think that the possibility of, and cure is a tough word, I would say long-term clinical complete responses. We are starting to see that in diseases we never thought we'd see it before, so I don't know why we can't do it in breast cancer.
Thank you, Dr. Yee. So our next question is, why do some physicians rely on tumor markers when others do not?
Yeah, so the tumor marker question. I will give you a history of the tumor marker question, a little bit of history. So we understand that cells will shed things into the bloodstream. And some of those, the tumor markers, are antigens that are expressed on cells, and they're shed into the bloodstream, and we can detect them. First, not all women have tumor markers, even though they might have metastatic disease. Secondly, some patients have elevated tumor markers and don't have metastatic disease. So in general, for following patients, and particularly following patients after a course of adjuvant therapy, they're not recommended. In patients with metastatic breast cancer, I actually follow them, and I use them as kind of a surrogate marker for how well we're doing at suppressing tumor growth. Now, I realize it's not perfect. Everybody recognizes it's not perfect, but in terms of testing, it's easier than some other things that we do. Now, I will comment, and I didn't talk about this. I had a section, but I think we are entering an era now where we can be much more sophisticated around tumor markers than we have been so far. The technologies that I showed you to measure DNA, we can now start measuring cell-free DNA in the bloodstream. We can start measuring circulating tumor cells. We can start measuring little chunks of cells that might have fallen off and are floating around. And whether or not those have benefit in the clinical management of breast cancer patients remains to be seen. I'm optimistic that we will come to a diagnostic test or a platform that will be much more useful to decide new therapies or new strategies than we have now. You probably know right now, many of your physicians might rebiopsy something to look for changes in any of the markers. And that's the best we have. But I think we will get there with circulating diagnostic tests as well. But I think we're a little bit off. So personally, I measure tumor markers because I think they're useful in some patients, but I understand why some other physicians may not. But particularly in the adjuvant follow-up setting, it's really not recommended.
Thank you, Dr. Yee. So our next question is, for someone with ER-positive breast cancer whose cancer progresses after taking multiple hormonal therapies, do they have any other options besides IV chemotherapy?
Personally, I like oral. If the decision is to go to oral chemotherapy, I think oral chemotherapy, Xeloda or capecitabine, is a very good option for estrogen receptor positive patients who have progressed through other options. First, it is for those women who have taken Xeloda. You can speak to this better than I can, but certainly in some aspects of what we worried about chemotherapy, you don't experience that with capecitabine, hair loss. Don't see a lot of nausea, vomiting. Don't see really white count suppression. Don't have to come into the clinic and you get an IV infusion. And in terms of choosing the first chemotherapeutic agent, I think capecitabine, Xeloda is a really good option. Now, it does point out a frustration I have is I don't know. I just said that, but I don't have any proof for an individual patient sitting in front of me that that statement is true. You know, we tend to go by our best judgment. What I mean is we don't have any predictive biomarkers that say, oh yeah, your tumor is extraordinarily sensitive to paclitaxel, Taxol, and it really doesn't care all that much about eribulin or Halaven. So I'm going to give you what your tumor is sensitive to. We're going to move that way. When I talked about the idea of we can measure blood, it's frequently called liquid biopsy. That's the hope everybody has, that we'll be able to make better informed decisions about what treatment to go to next.
Thanks, Dr. Yee. So one question that we have gotten is, when should someone consider genomic testing of their metastatic tumor? So
I don't know, can I just, anybody gotten their tumor sent off to Foundation or Claris? Okay, so a couple comments about that. Foundation, the current genomic testing mechanisms are really looking for mutations. And in breast cancer, mutations, actionable mutations, as we call it, is not necessarily frequently discovered in breast cancer. But we all hope it will be. And there actually is a national clinical trial called the NCI MATCH trial, where patients with metastatic disease are biopsied, have their tumor sent off, sequenced, and there's a list of roughly 22, 25 drugs that are targeted against specific mutations. Then if you match, you will then be assigned that drug. So this is the test of genomic testing that actually I think is probably going to result in actionable mutational items. But as a regular clinical practice now, it's not quite settled. I will say the NCI match is not open to it's open to all breast cancer. It's not just any specific cancer. And the match rate is somewhere south of 10%. In other words, the patients who send in their tumors, we only have drugs that match for 10%. And I think, I'm guessing it's even lower in breast cancer. I'm guessing it's really low in estrogen receptor positive breast cancer. But having said that, if that opportunity is available to you and your physician brings it up, I would certainly ask you to consider participating. Unfortunately, it does require a biopsy. But on the other hand, I think by accumulating genomic information, we'll be able to make better decisions.
Thank you, Dr. Yu. We have a lot of questions about genomic testing and blood biopsies. I just want to let the audience know that Living Beyond Breast Cancer is going to have a whole program on this a week from Monday. So we'll give you more information about that. So we are going to take 2 more questions. One person is asking if they have been NED for a couple of years or longer, how often should they be getting monitored, getting scanned, so on and so forth?
Yeah, boy, that's a tough question. For those patients who see me, you know I'm not a big fan of lots of testing. I'm a big fan of lots of testing or anatomic testing, scanning, if something is different. Something is different in the way you feel, something is different in any of the blood tests. And so if someone's been NED for really prolonged periods of time, I tend not to do it unless something is completely different. I'm just thinking one patient in particular that has been NED after Kadcyla for a long, long time now. And we talk about this every visit, and I basically kind of say that I don't know how much better you'll feel if we do the scanning. But it's an individual decision I think patients need to have with their physicians.
Thank you, Dr. Yu. So we'll have one more question before we unfortunately have to stop. We have a number of questions from folks whose primary breast cancer was hormone receptor positive, but at their metastatic recurrence was triple negative, and wanting to know what's the best strategy for treating that type of breast cancer.
Yeah, so we don't really have actually a very good answer for why that happens. Just to think about it conceptually, it's possible that the tumor lost ER expression over time, or it's possible that there was a mixed set of populations at the time 0, or it's possible that if you ever look at a path report of primary breast cancer, they will give you a percentage of cells that are ER positive, and very often it's not high, but we still treat those patients with endocrine therapy, and there's been shown to have benefit. But it's very possible we selected out the ER positive patients. So at any rate, that was a long way around getting to the question. If it truly is triple negative and not displaying any of the markers, then I think thinking about chemotherapy is valuable. I will say something that's off-label. The off-label is I've been asking our pathologists to measure androgen receptor more frequently than before in triple negative breast cancer. It happens to be we have a trial for an androgen receptor antagonist, but I do know that there is, and the reason I'm saying this off-label because it's not completely settled yet, that how valuable androgen receptor is as a target in triple negative breast cancer. But that's an option to have a discussion with your physician. If you were androgen receptor positive, is there a trial for you?
Thank you, Dr. Yu. Can we all give him a hand and thank him?